This is Lecture 14, The Wonderful World of Complement, Part 1. So I'd first like to begin with this complement series and give you this case study. So a 21-year-old female presents to the ER with severe joint pain, increased temperature with visible and notable skin lesions on her hands and feet, increased white blood count. They did a scraping of the pus from one of these lesions and grew Neisseria gonorrhoeae, and they did some chemistries at CH50, which was 0, at elevated C3, C4, C5, and factor B. And so as we go through these next couple of lectures, please keep this case study in mind, and we'll kind of come back to this after the end of these lectures, and we'll kind of go back over this case study. So in this lecture, we're going to be discussing the complement activation pathway. So there are 3 pathways that we'll be talking about: the classical pathway, the alternative pathway, and the lectin pathway. And we'll be able to differentiate between these 3 different pathways. And then we'll break down the different components of proteins that are involved in the complement cascade. So a brief medical history. This will not be on the exam, but it is relevant to the material that we'll be covering. In early 1890s, Hans Ernst August Buchner was the first person to describe a substance. He didn't know exactly what it was, but he found that a substance in the blood was actually capable of destroying bacteria. And later in 1896, Jules Bordeaux determined the substance had actually 2 components to it. So one of the components is that it maintains this destroying bacteria effect after being heated, and another component actually loses this effect after being heated. And in the late 1890s, I'm sure after 1896, Paul Ehrlich determines the heat-stable component is antibodies, and the heat-labile component is complement. So when we talk about heat-labile proteins, these type of proteins are the ones that can be changed or destroyed in high temperatures or high heat. So heat-stable means that they can withstand to a certain degree high temperatures, and heat-labile is that they can be destroyed at high temperatures. So most of these heat-labile plasma proteins are made by the liver. Alright, so one thing throughout this program, you could pretty much guarantee that if we ask you where a protein is made, it's usually made by the liver. Probably 90% of the time, the liver makes all the proteins. Unless you know 100% sure that it is not made in the liver, I would guess the liver. So these functions of these heat-labile plasma proteins, alright, they can be opsonins, alright. They can help with complement-mediated cytolysis, help with phagocytosis. They can help amplify the inflammatory response, and they also assist in removal of some immune complexes. And these immune complexes meaning antibodies attached to a certain cell or a virus or what have you. So immune complexes, when we talk about immune complexes, we're actually talking about something that an antibody is attached to. Okay. So before we dive into the pathways, we first need to learn what the pathways are made up of. Alright, their components and how these components are named. So the first way that they are named is alphanumeric. Alright, so we have our C1 through C9, our complement component 1 through 9. Alright, we could also use letters like factors B and factors D. And we could also use common names like propertin, which is factor P. I know medical terminology is not always cut and dry, so I will try to help you along the way as I'm naming these off when we're going through the pathways. Also, as we talk about the pathways, we're going to be seeing some of our complement components get cleaved or fragmented. Okay, so for example, we have our complement component C3, and this C3 is going to be fragmented or cleaved into C3A and C3B. So this one C3 protein will then be cleaved into two different proteins, C3A and C3B. Now, each one of these proteins will have a different effect on each one of these will have a different purpose. Okay, so for instance, C3A typically is the smaller fragment, and it usually diffuses away. So C3A diffuses away from the target that we're trying to destroy. C3B is the bigger fragment and usually binds to the target cell that we're looking at destroying. This is usually the case. C3A usually diffuses away, and C3B usually binds to the target. And we could kind of take this general idea with all the other complement components, say C2, etc. But there are some fragments or some components that doesn't follow this particular rule. So when we talk about those, I'll be sure to make note of that to you. As we go through the pathways as well, we'll also see fragments that actually interact with each other to form complexes, and these complexes have enzymatic capabilities. For instance, C4B2A can cleave C5, alright, into C5A and C5B. And these enzymatic complexes are denoted with a bar over the top of them. So when you see that bar, you'll know that this is a complex and it does have enzymatic activity. So in all three different pathways, the classical pathway, the alternative pathway, and the lectin pathway, one major complement component plays a vital role in all three pathways, and this would be C3. So the first pathway that we're going to be discussing is the classical pathway. Majority of the time, the classical pathway is going to be activated by an antibody-antigen complex. So typically antibodies are needed to start the classical pathway. However, there are a few instances that CRP, C-reactive protein, and a few other microorganisms can activate the classical pathway, and this typically during this activation with CRP or microorganisms, an antibody is not needed to start the classical pathway. But probably 99% of the time, antibody is necessary or is used in starting the classical pathway. As we have learned in the last few lectures, all antibodies are not created equal. So the antibodies that could actually trigger the classical pathway are IgG1, 2, 3, and IgM. And obviously IgM, being the bigger of the antibodies, is more effective at triggering the classical pathway. IgG needs 2 IgG molecules or antibodies close together in order to initiate the start of the classical pathway, so it needs at least 2. IgM obviously only needs 1 antibody to trigger the classical pathway. Alright. So for instance, we could say in order to lyse a red blood cell, we only need 1 IgM. For IgG, we need 1,000 IgG antibodies to lyse 1 red blood cell. And this is mainly because the antigens are so spread apart on a red blood cell that we have to have 1,000 linked together in order to start this classical pathway, this complement cascade. Alright. So the classical pathway is a multi-step process. So when we go down this pathway, we're going to be coming across many amplification steps. So it's actually building on itself. Okay. So we have our C1 through C9. So these are our complement components. One thing you want to remember, and please make note of, is that we have C1 through C9 with 4 out of line. So remember that I told you these were numbered when they first were discovered. So first one was discovered was C1, then C2, and then C3. Then after they discovered C3, they discovered C4, but it was actually towards the beginning of the process. So that's how we say C4 is out of line. So we have C1, C4, C2, C3, C5, 6, 7, 8, and 9. Now, the classical pathway and these complement components are divided into 3 main stages. We have our recognition unit, our activation unit, and then our membrane attack complex. And we'll go over each one of these. So the first stage is the recognition unit. So this recognition unit is made up of C1. So C1 will bind to the Fc region of the antibody, and once it binds to this Fc region, it will become activated. Now C1 recognizes the CH2 region on IgG. Okay, that's the CH2 on the constant portion of the antibody, and it recognizes the CH3 region on IgM. So C1q will bind with C1R and C1S and will become activated. Alright, so we are going to actually have our first enzymatic complex when these three combine. So our first stage is our recognition unit, our C1. So we have our inactivated C1QRS or C1 complex, and it will come in and connect to the FC portion of our antibodies. Let's say these are IgGs, okay? So this C1 complex binds to the FC portion of our IgG, then another C1R and C1S will come and bind to this C1 complex. Now these C1S and C1R that comes in and binds to the C1 complex are activated, and this will create an enzyme, our first enzymatic complex. And this enzymatic complex is named C1QR2S2. Alright, so basically, to break this down, we have our C1QRS complex, then we're just adding one more C1S and one more C1R, and this is how we get C1QR2S2. And this is our first enzymatic complex in the classical pathway. So the next stage in our classical pathway is the activation stage, or our activation unit. And this activation unit is comprised of C4, C2, and C3. So we have our C1 that is attached to our FC portion of our IgG antibodies. And we have C4 floating by, and C1 will cleave C4 into C4A and C4B. So these are the two fragments that C4 is cleaved into. C4A will diffuse away, and C4B will bind to the cell membrane. And we consider this to be our first amplification step. And after that, C2 will come floating by, and C1 will bind or will cleave C2 into C2A and C2B. C2, however, is the black sheep of the group. So remember that I said that typically A will diffuse away and B will bind. Well, C2 does not adhere to that rule. So C2A will bind to C4B, and C2B will diffuse away. So this will leave us with our first enzymatic complex. So our first enzyme complex is C4B2A, and that is denoted with that bar over the top of that name. Now it's also called C3 convertase. So C4B2A enzyme is C3 convertase. This diagram is taken from your immunology textbook. We have our C1 that is bound to the FC portion of our IgG molecules. Alright, so we will have C4 that comes along. C1 will cleave C4 into C4A and C4B. C4A will diffuse away, and C4B will bind to the target cell. C2 will come along, and C1 will cleave C2 into C2A and C2B. C2A will bind to C4B, and C2B will diffuse away. So now we have our C4B2A enzyme complex, and this is also called C3 convertase. One thing to make note of, this will not be on the exam, is that when C1 cleaves C4, C4B only has a few seconds to bind to the cell membrane. Otherwise, it will become inactivated, and it could potentially bind with water molecules that will inactivate this C4b. So we have our C3 convertase, which will cleave C3. So it'll cleave C3 into C3A and C3B. C3A will diffuse away, and C3B will bind to C4B, C2A. Alright? So C3B is what we consider a very powerful opsonin, and we will definitely talk more about C3B in the coming lectures, but just remember that C3B is a powerful opsonin. So we have our C3B that binds to C4B, C2A, and this will form a new enzymatic complex. So we now have our C4B2A 3B enzyme, and we call this C5 convertase. And what do you think C5 convertase is going to cleave? So these two diagrams are also found in your textbook. So we have our C3 convertase in our top left diagram. C3 will come along. It's going to cleave C3 into C3A and C3B. C3A diffuses away, and C3B will bind to this C3 convertase, which will then have a new enzyme named C4B2A 3B, or C5 convertase, which will then cleave C5. And once we have this C5 convertase, we will then start to get into our membrane attack complex, also known as MAC. So our C5 convertase will cleave C5 into C5A and C5B. C5A will diffuse away, and C5B will attach to the membrane. And then C6 will come along, and C7 will come along, and C8 will come along and bind to this C5. And this will create our next enzyme, our C5B678. And at this stage, this enzyme can lyse a red blood cell, but it cannot lyse a nucleated cell. That is very important to remember. So at this stage, this enzyme C5B678 can lyse red blood cells, but not nucleated cells. So this is a diagram from your book as well, and we have our C5B complex, and then we will have our C6, 7, and 8 come down and bind to this C5B to create our C5B 6, 7, 8 enzyme. And at this stage, remember, it can lyse red blood cells, but it cannot lyse nucleated cells yet. So the last one that we're waiting for is C9. So this is our last complement component that we are actually waiting for to have a complete cascade. So C9 polymerizes. So we'll have multiple C9 molecules connect to this C5b678 complex. So once we have this C9 attached to this C5b complex, it is now able to punch holes into the cell membrane, and thus it can destroy a nucleated cell through what we call multiple hits. Okay, so it will punch through the cell membrane of the cell, and this will create an influx of water, loss of electrolytes, and basically make the cell inoperable. And here is another diagram of this C5b complex. So when C9s come along, typically there's around 10 to 17 C9s, and they will start punching holes into the cell membrane, and this is what creates the influx of water and loss of electrolytes within the cell. And once this happens, the cell is no longer viable. And here is an electron microscope image of what complement could actually do to a bacterial cell. So you can see all the holes that it has punched into this bacterial cell. So let's take a final look at our classical pathway. So we have our recognition unit of C1, which will attach to our Fc portion of our antibodies. Then we will get into our activation unit. So C4 will come floating by, and C1 will cleave C4 into C4a and C4b. C4a will diffuse away. C4b will bind to the target cell membrane. C2 will come along, and C1 will cleave C2 into C2a and C2b. C2b will diffuse away, and C2a will bind to C4b, creating our second enzyme. And this is C4b2a, also known as C3 convertase. C3 convertase will then cleave C3 into C3a and C3b. C3a will diffuse away, and C3b will bind to our C4b2a. And this will create C4b2a3b enzyme, also known as C5 convertase. Alright, now our C5 convertase will then cleave C5 into C5a and C5b. C5a will diffuse away, and C5b will attach itself to the target cell membrane. Then we will have our C6, 7, and 8 come by and attach itself to our C5b, and this will be an enzyme. And at this stage, remember that it can actually lyse a red blood cell, but not a nucleated cell. And finally, our C9s will come along. Remember, around 10 or 17 C9s will come along and join to C5b, 6, 7, and 8. And at this stage, it can lyse any cell. So let's take this moment to talk about the A and B properties. When we cleave a complement component, we have, for instance, in this example, we have C3a and C3b. So C3a, we consider this to be an anaphylotoxin. Okay, so once C3 is cleaved, C3a now has specific duties that it can do. It will diffuse away and start to help the inflammation process in other ways. So it can help in the release of histamine. It can help with dilating the blood vessels, contract smooth muscles, and this all in turn allows for vascular fluid leak. So increase the fluid to the site of the response. C3b is considered to be a strong opsonin, and we will go into great detail about C3b, especially in the other pathway, the alternative pathway. Alright, so C3b is considered a strong opsonin. It can bind to CR1 receptor on cells. Alright, and there is actually an inactivated C3b form. This is IC3b. Now, factor I inactivates C3b, and this is how we get the IC3b. And we'll discuss this later as well. So, IC3b is also an opsonin. So, how can an inactivated form of C3b be an opsonin? So, on B cells actually has a receptor for C3b or IC3b as well. So once this IC3b binds to this B cell, it then becomes an opsonin. So what is an opsonin? Remember that opsonins help phagocytosis. So this will allow the B cell to actually bind to bacterial cells, etc. So this is just a little diagram of C3b and CR1 receptor on a phagocyte, etc. So C3b can actually attach to a bacterium. Alright, so we have C3, and we'll talk about this in the next pathway. We have C3 floating throughout our circulatory system all the time, and C3 will spontaneously hydrolyze, which we'll talk about as well, and cleave itself into C3a and C3b. And this C3b, remember, is a strong opsonin. So the C3b will attach to bacterial cells, and then a phagocyte like a macrophage or a neutrophil will come by, and they have these CR1 receptors on their cells. And the C3b and CR1 will match up. Alright? And this is what creates this opsonin, this intense attraction, and then this phagocyte, whether it be a neutrophil or macrophage, will engulf that bacterial cell. So C3b plays a tremendous role in opsonization and control of bacterium, etc. in our bodies. So when we say that a component is an anaphylotoxin, they all have the same properties, or they're all able to do the same thing. So the contraction of smooth muscle increases vascular permeability and can cause histamine release from mast cells, etc. So all these anaphylotoxins have this capability. Some are better at it than others. For instance, C4a is weaker than C3a and C5a, so we don't have that big of histamine release from mast cells, for example. C5a is a major attractant and a strong anaphylotoxin, like C3a is. Okay, but C5a actually has a better opportunity or better job at PMN aggregation or bringing neutrophils to the site of where it was cleaved. It also has the ability for lysosomal enzyme release, all right, and TNF alpha, tumor necrosis factor alpha release. So it triggers macrophages and neutrophils to come to the site and start to release some of these enzymes. So it plays a major role in the inflammatory process. Here's a quick review. If you'll answer the questions, we'll go over the answers on the next slide. So which antibodies fix complement? That would be IgG and IgM. Remember IgG 1, 2, and 3. Which is the best? IgM. And what is the basic sequence in the classical pathway? That would be C1 through 9 with 4 out of line. Which component is considered to be the recognition unit? That would be C1. Which components are in the activation unit? That would be C4, 2, and 3. And which components are in the membrane attack complex, or MAC? That would be C5, 6, 7, 8, and 9. So let's get to our second pathway, and this is our alternate pathway. So in our alternate pathway, we do not need an antibody to trigger the start of this pathway. It can be activated by a number of different substances, and it's typically activated by bacteria containing lipopolysaccharides on their cell membrane. All right, and we consider this alternate pathway a component of our natural defenses. This alternate pathway also has been known as the Properdin system or Properdin pathway. That term is usually not used anymore. We usually call it the alternate pathway. And what's key to note about this is that C1, C2, and C4 are not involved in the alternate pathway. All right, remember that we do not need an antibody to activate this pathway. So C1, which connects to the antibody and starts the classical pathway, we don't have antibodies to start this pathway, so we will not need C1, C2, or C4. So remember when I was talking previously about C3 spontaneously hydrolyzing. So we have C3 constantly in our serum circulating throughout our bodies, and it goes through a process called tick over, what we call tick over. So it spontaneously hydrolyzes and it forms C3b. So C3b is flowing throughout our system as well. So remember, C3b is a strong opsonin. So what stops this C3b from landing on our host cells and preventing our host cells from being destroyed? What prevents this? So our host cells actually inactivates this C3b due to sialic acid. All right, so this sialic acid actually protects our host cells from accidental activation of the alternative or alternate pathway. Now, C3b will bind to foreign substances, foreign cells, because these cells actually have low levels of this sialic acid, and C3b will bind to normally polysaccharide surfaces of the bacteria cell. So this is what will then activate the alternate pathway. Okay, so we have our C3b that has found and deposited on a bacterial cell surface. All right, so we have our C3b that's already on the surface. Factor B will come along and bind to C3b. Okay, so now we have C3b, big B. All right, big B meaning factor B. Then factor D will come along and cleave factor B into factor B A and factor B B. Now, once factor D cleaves factor B, we will then have our first enzyme. We have our enzyme C3b, big B, little b. And this is also called C3 convertase. All right. One thing to remember about this C3 convertase is that it has an extremely short life. It can be disassociated by the action of factor H. So if C3 does not come along and be cleaved by the C3 convertase, then factor H can come along and disassociate this, and will the pathway will then be mute. It will stop at that point. So as long as factor H doesn't disassociate our C3 convertase, then properdin will come along and help stabilize this enzyme. All right, so this is where the properdin system came about. So we have our properdin that is stabilizing this C3 convertase, and then C3 will come along, and this C3 convertase will cleave C3 into C3a and C3b. C3a diffuses away as an anaphylatoxin, and C3b will bind. So now this will create our new enzyme. We now have C3b, big B, little b, 3b. And this is called C5 convertase. And then the subsequent steps are the exact same as the classical pathway. So this is a diagram taken from your textbook. So we have our C3 that will spontaneously hydrolyze and have become C3a and C3b. C3b will deposit onto the bacterial cell wall, and then factor B will come along and bind to C3b. Then factor D will come along and cleave factor B, and so we will get our first enzyme, our C3b, big B, little b. Okay. Then properdin will come along and help stabilize this enzymatic unit. And then another C3 will come by, and it will, this C3 convertase now that we have will cleave C3, and then we will have C3b, big B, little b, 3b, and this will be C5 convertase. And then C5 will come along, the C5 convertase will cleave C5 into C5A and C5B. C5A will diffuse away as an anaphylotoxin, and C5B will get deposited on the cell, and then C6, 7, and 8 will come in and bind to C5B, and at that point, remember that it can lyse a red blood cell, but this is not a red blood cell. So then C9 will come along and start to actually punch holes into the bacterial cell wall. So here's a quick review over the alternate pathway. If you'll take a few minutes and answer these questions, we'll go over the answers in just a second. So the first one is define takeover. So it is when C3 spontaneously hydrolyzes or spontaneously cleaves. So C3 will cleave into C3b and C3a. So what activates the alternative pathway? Most of the time it is microorganisms. What four serum proteins are involved in the activation of alternative pathway? So we have C3, factor B, factor D, and properdin. What is the function of properdin? It is a stabilizing, has a stabilizing effect on C3b, big B, little b, or C3 convertase. Where do the classical and alternative pathways intersect? And that would be at C5 convertase. The last pathway is the lectin pathway. And this pathway provides a link between the innate and acquired immune response because it involves nonspecific recognition of carbohydrates that are common constituents of microbial cell walls, and they are really distinct from those found on human cells. So this pathway is mainly triggered by the binding of polysaccharides to circulating lectins in our bodies. And these lectins are proteins that actually bind to carbohydrates on the cell walls of these invading pathogens. These invading pathogens can be bacteria, viruses, yeasts, and even some parasites. So the lectin pathway is activated by mannose residues or certain types of sugars found on the bacterial surfaces. So the key difference between the lectin pathway and the classical pathway is lectin pathway uses mannose binding lectin. Okay, so this is a lectin that is binding to the mannose on the bacterial cell. This mannose binding lectin, or MBL, is very structurally similar to the C1QRS in the classical pathway. This MBL is composed up of 3 enzymes called MASP1, MASP2, and MASP3. Alright, MASP2 typically takes the active role in cleaving C4 and C2, and once this cleaving occurs, then the subsequent steps are exactly the same as the classical pathway. So the really only difference between the lectin pathway and the classical pathway is that the lectin pathway uses a mannose binding lectin. Okay, and it will bind directly to the bacterial surface. So the lectin pathway does not need an antibody to start the pathway. So here's a diagram of the lectin pathway. So we have our MBL that has attached itself to the pathogen surface. Alright, and then C4 will come along. This MBL will cleave C4 into C4A and C4B. So C4A will diffuse away, C4B will attach itself to the pathogen surface. C2 will come along, and MBL will cleave C2 into C2A and C2B. C2B will then diffuse away, and C2A will then bind to C4B, which creates what? C3 convertase. And then the subsequent steps are identical to the classical pathway. Okay, so let's review the lectin pathway. Answer these questions and we'll go over the answers on the next slide. So what activates the lectin pathway? Polysaccharides that bind to the lectin. Alright, what are lectins? They are proteins that bind to the carbohydrates. And what is the primary lectin involved in activation? That would be the MBL, or mannose binding lectin. So where does the lectin pathway intersect the classical pathway? That would be at C3 convertase. And here's a little diagram of the 3 different complement pathways and how they intersect with each other. This is another diagram of the complement pathways. This is taken from your textbook, and it is in the 4th edition. It's located on page 98. This is the end of part 1 of complement. We will be going over part 2 in the next lecture.